Bismillahirrahmanirrahim. Good morning, dear students. Today we are going to discuss an important topic. It is about staphylococci. We have said before that bacteria are classified into gram-positive and gram-negative according to the structure of their cell wall and the uptake of the gram stain. And each group is further divided into gram-positive cocci and the bacilli, gram-negative cocci and the bacilli. Today we are going to discuss a type of the gram-positive cocci, which is the Staphylococcus. The morphology of Staphylococci, as you see here, it is characterized by being gram-positive cocci. It is arranged in clusters. These clusters are meaning groups like grapes. It is non-spore forming. As we said before, the spore is a form of bacteria. When the conditions surrounding the bacteria are unfavorable, it is a way of resisting to these unfavorable conditions to change into a spore. Once the conditions are improved and nutritional requirements are available for the bacteria, it can regerminate into the vegetative form, producing its pathogenic effect. This criteria is not present in Staphylococci. It is non-motile bacteria. It has no flagellae. Some strains of these bacteria can form a single simple layer surrounding it, which is called the microcapsule. Others can be surrounded by a less developed, very thin layer, which is called the slim layer. We classify the staphylococci into coagulase positive staphylococci, which are Staphylococcus aureus, and coagulase negative, which includes Staphylococcus epidermidis and Staphylococcus subtilis. Let's start by Staphylococcus aureus. About 10 to 40% of normal individuals who are not suffering from any disease are considered carriers for Staphylococcus aureus, which is present in the anterior nerves. The danger of such people that they shed these organisms which can be transmitted to others, causing pathogenic diseases in them. But these individuals acting as carriers are not showing any disease or any evidence of having this bacteria. Staphylococcus aureus is a very important and common pathogen. What about the virulence factors of Staphylococci? Staphylococci have different factors that help it to produce its pathogenic effect over the human being, including enzymes. The types of the enzymes which are produced by Staphylococci are: number one, the coagulase and the clumping factor. What is the importance of the coagulase or the effect of this coagulase enzyme? It converts the prothrombin into thrombin that will act over fibrinogen to change it into fibrin. What about this fibrin? It will coat the bacterial cell. You know that one of the mechanisms of the immune response was the opsonization and phagocytosis, where the microbe or the bacterial cell was coated by immunoglobulins and complement proteins. And such opsonins, which are the immunoglobulins and the complement proteins, are having receptors over the phagocytic cells. So when the bacterial cell is coated by the immunoglobulin and the complement proteins, it enhances and facilitates the process of the phagocytosis. The presence of fibrin around the bacterial cell hinder the binding of the complement and the immunoglobulin, and so will affect the opsonization and the phagocytosis. Other enzyme is catalase enzyme. This enzyme has an effect over the hydrogen peroxide, breaking it down into oxygen and hydrogen. The mechanism of killing of the bacteria inside the phagocytic cell depend on the presence and the effect of hydrogen peroxide. So when the bacteria can produce catalase, it can prevent the effect of this hydrogen peroxide, maintaining the survival of the staphylococci in the phagocytic cell. Other enzymes like proteases, lipases, and deoxyribonucleases acting on proteins, lipids, and DNA. What about the exotoxins? These are toxins which are secreted by this bacteria to diffuse through the body, damaging different structures inside it. The first type of the exotoxin is the membrane damaging toxin. as hemolysin that act on the RBCs producing their lysis, and leukocidin that act on the leukocytes producing their damage. We have 6 different enterotoxins which are produced by Staphylococcus aureus. Enterotoxin means that it's a toxin that acts on the GIT. The different types are named A, B, C, D, E, and G. They produce food poisoning. The criteria of these toxins is that they are resisting the effect of heat and acid. Another important toxin is the toxic shock syndrome toxin. This toxin produces a specific disease which is called the toxic shock syndrome. The last type of the toxins is the epidermolytic or the exfoliative toxin, a toxin that produces severe damage to the epidermis of the skin and produce a disease which is called the scalded skin syndrome that usually affects newly born babies. What about the diseases which are caused by Staphylococcus aureus? We categorize these diseases into 2 groups. The first one is suppurative or disease characterized by production of lots of pus, and according to the site of infection, the clinical presentation differs for each patient. For example, it can affect the skin, affecting the sebaceous glands of the skin, or affecting the hair follicles of the skin to produce boils or styes or furunculosis. Sometimes it causes invasive infection affecting deep organs inside the body. If it affects the lung, it produces pneumonia, the CNS, meningitis, the urinary tract, it produces urinary tract infection like nephritis or glomerulonephritis or urethritis or urinary bladder infection. It can be present in the blood to cause bacteremia or septicemia, and it can produce endocarditis. What about the diseases which are caused by the toxins produced by the bacteria? They are called the toxogenic diseases. They include scalded skin syndrome, food poisoning, and toxic shock syndrome. The scalded skin syndrome, as we said, it's a disease which is caused due to the production of the epidermolytic or the exfoliative toxin. This toxin produces lysis and shedding of the epidermal layer of the skin. So the baby is characterized by widespread blistering and losing of his epidermis. As we said, it usually occurs in newly born babies. Another disease is the food poisoning. It is caused by the enterotoxins, which we said that there are different 6 types of enterotoxins produced by Staphylococcus aureus. They are characterized by being resistant to heat and acid. The commonest type of food that is liable to be contaminated by the toxins of the staphylococci is the milk products when the milk used in preparing them is not well pasteurized or well sterilized. The incubation period is usually 1 to 8 hours, and then the patient will develop nausea, vomiting, and diarrhea. What about the toxic shock syndrome? This is a disease which is common in the females that use tampons. When the tampon is inserted inside the vagina for a long period of time, it will stimulate the colonization of the vagina by the Staphylococcus aureus. Following the colonization, these staph will start to produce an important exotoxin which is called the toxic shock syndrome exotoxin. As you know, usually this bacteria when present, it will shed antigens, and the antigens will be caught by MHC class 2 on the antigen presenting cell to be presented to the T cells through the T cell receptor. This interaction will lead to production of different types of cytokines like interleukin 2, interferon gamma, interleukin 1 beta, tumor necrosis factor alpha that will lead to the immune response. But as we have known before, after the destruction and the killing or elimination of the antigen, this reaction should stop and the immune system should stop producing the different types of cytokines. But in case of presence of the toxic shock syndrome toxin, it is called a super antigen. What is the meaning of a super antigen? An antigen that stimulates continuous stimulation of the immune system. So the presence of this super antigen will keep the interaction between the MHC class 2 over the antigen presenting cell and the T cell receptor over the T cell without ending. So the cell is continuously activated, producing more and more aggressive amounts of the cytokines. The production of large amount of cytokines in this case is not beneficial, as it will lead to severe damage in different organs of the tissue, resulting in the manifestation of the toxic shock syndrome, which is fever, vomiting, diarrhea, desquamation of the skin, hypotension, and finally can lead to death. How can we diagnose a disease that is caused by Staphylococcus aureus infection? As we have known before that any disease to be diagnosed, it needs to take the proper sample. We choose the sample according to the site of infection. For example, if we are having skin manifestations, we can take a swab, we can take pus, skin lower respiratory tract infection, we take sputum, meningitis, CSF, bacteremia or septicemia, blood. The first step for the sample is to carry out direct film. And we said before that the direct film can be carried out stained and unstained. Unstained to detect motility, and as we said, staph is not motile. So there is no need for the unstained direct film in this case. We depend on the stained direct film. By staining in gram, we can see these are the cells in the sample, and these are the staphylococci, which is characterized by being gram-positive cocci and arranged in clusters, non-motile, non-sporing, and capsulated with a slim layer or microcapsule. What about the culture? We should discuss the culture characters. Regarding oxygen requirements, it's facultative in Europe. The suitable temperature is 37 degrees Celsius. The amount of CO2 that present in the atmosphere is sufficient to support the growth of Staphylococcus aureus. The types of the media used, it can grow over the ordinary media and to produce a specific type of pigment which is golden yellow in color. It can grow over the blood agar and produce beta hemolysis. We can use a selective medium which is characterized by having a component that stimulate the growth of certain type of bacteria and inhibit growth of others. In this case, the presence of the mannitol is characterized that it supports the Staphylococcus aureus growth through fermenting this mannitol, and the presence of salt will inhibit other bacteria present in the normal flora other than Staphylococcus. This is the nutrient agar plate or the ordinary media with the golden yellow colonies of Staphylococcus aureus that are produced due to the production of the endopigment. And again, if you remember, we have two types of pigments, endopigment and exopigment. The endopigment remain localized inside the colony of the bacteria, giving it its characteristic color without affecting the media, and the exopigment that usually diffuses outside the colony, giving the color to the media, and the colonies appear pale or colorless in color. This is the mannitol salt agar with the colonies of Staphylococci over it. As a result of the fermentation of the mannitol, a change has taken place in the color of the media, and a specific appearance of the colonies will appear, indicating the growth of Staphylococcus. How can we identify the colonies? First, from their appearance over the media, like the production of the beta hemolysis, the production of the endopigment, which is golden yellow in color, or the characteristic appearance over the mannitol salt agar. Then we make another film from the culture to detect the morphology when stained by gram. Again and again, it's gram-positive cocci arranged in clusters, non-motile, non-sporing, having a slim layer or microcapsule. Then we carry out biochemical reactions. The most important biochemical reactions is the coagulase test to differentiate it from the coagulase negative Staphylococci and to detect that it is Staphylococcus aureus. Then we use the catalase test, and if you have studied before the group of another group of gram-positive cocci, which was called the streptococci, that can be differentiated from the staphylococci by the effect of the catalase test. We said that the catalase act on hydrogen peroxide, breaking it down into oxygen and hydrogen. So this is a slide containing a drop of hydrogen peroxide, and we take a colony from the staphylococci to put it inside this hydrogen peroxide. This bubbling indicates the release of catalase enzyme, the breaking down of hydrogen peroxide, and the formation of hydrogen and oxygen. So this is a positive test. Nothing happened here, indicating that no catalase enzyme was produced. It's a negative test. And finally, the fermentation of the mannitol can be used to identify the Staphylococcus aureus. What about the phage typing? Is a type of virus which is called the bacteriophage. You know that viruses usually infect human, infect animals, infect plants, but a certain group of viruses infect bacteria. They are specific to certain types of bacteria, and even the different types of phages can be used to differentiate between different strains of bacteria. So in this plate, we have inoculated the strains of the bacteria, and then we put a drop of each type of phage over each square. The site or the square where the lysis takes place indicate that this bacteria was sensitive to this phage. And we have already known the different types of the phages and the sensitivity of these phages and specificity to different types of bacteria. So we can use this phage typing for the identification of the type of bacteria. What is MRSA? It's a type of staphylococci that is highly resistant to antibiotics. You know that due to the misuse of antibiotics, it led to the development of different strains of bacteria that are resistant to antibiotics. One of these strains is the methicillin resistant Staphylococcus aureus. It's a type of Staphylococcus aureus that produce enzymes that break down methicillin antibiotic. This type of bacteria is treated by vancomycin, and it's a very dangerous type of bacteria and produce different types of infections. This is the staphylococci, the coagulase positive staphylococci, or the Staphylococcus aureus. Now we are going to discuss a different type of staphylococci, which is coagulase negative staphylococci. It includes two types. The first one is Staphylococcus epidermidis, and the second is Staphylococcus saprophyticus. The epidermidis is characterized by being a part of normal flora. It's present over the skin, and as it is a flora, it is non-pathogenic. But when can it produce disease? You know that flora, as long as it is present over its normal area, and the patient or the person immune system is intact, he is not immunocompromised, the flora cannot produce disease. But if the flora shifted from its normal place to another place, or the patient become immunocompromised due to any cause, in these conditions, the flora is no longer considered the flora and beneficial. It may become pathogenic and produce different types of infections. This is for the Staphylococcus epidermidis. What about the saprophyticus? Another type of coagulase negative staphylococci that commonly produce urinary tract infection in young males. How can we differentiate between the different Staphylococcus species? For the Staphylococcus aureus, we said that it's coagulase positive. It is also able to produce DNase enzyme, an enzyme that can break down the DNA of the infected cells. It can ferment mannitol, as we said. It produces different types of exotoxins. It produces hemolysin. And also it has an important antigen over its cell surface, which is called the protein A. This is specific for Staphylococcus aureus and absent in the epidermidis and saprophyticus. How can we differentiate between epidermidis and saprophyticus? According to their sensitivity to an antibiotic which is called the novobiocin. The staph epidermidis is characterized by being sensitive to novobiocin. Saprophyticus is resistant to novobiocin. How can we test it? This is a plate inoculated with the Staphylococcus epidermidis. This is another one inoculated with Staphylococcus saprophyticus. And this is the antibiotic disc containing novobiocin that was put over the surface of the plate after plating it with the bacteria. And here also it is put over the surface of the plate to be incubated in the incubator for 24 hours. Then, by examining the plate, we find that we are having an inhibition zone of growth of the bacteria surrounding this novobiocin disk, indicating that the epidermis is sensitive to novobiocin. But the saprophyticus, no inhibition zone is present, so it is resistant to the novobiocin. So this is the method that can be used to differentiate between its sensitivity and resistance to novobiocin. This is the end of our lecture. I hope that you understood the different types of staphylococci. How can you differentiate between coagulase positive and coagulase negative? And how can you differentiate between the two types of the coagulase negative? Wish you the best of luck, and I hope you study well and be the best. Thank you and goodbye, dear students.